Hello students, today's topic is biotechnology. Okay, this is very interesting topic actually. Nowadays these are the very important aspects of biology that is applied for human welfare. Okay, we are using biological organisms, biological species, okay, biosystems, organs, tissues, cells, okay, biomolecules means proteins, okay, carbohydrates, nucleic acids to enhance, to amplify, to make our life more comfortable, more enjoyable as well. Okay, so biotechnology, so this is very important today in today's life. Okay, so biotechnology, bio means whatever life forming, life forms, maybe it is organismic level or tissue level or cell level or biomolecular level means proteins. We can eat proteins also. Protein powder is there, in the market. Protein powder is there, protein component, protein only protein component is there. Okay, so we can eat that also. So bio means bio life forms. Okay, and technology, whatever science is there behind the technology, we can apply those into in the biological species or biological systems to get some benefit for our life. Okay, for our life processes. Okay, so first of all, so principles, actually what we do, whatever the technology or whatever the knowledge we have in the engineering discipline, okay, or some technological advancements, we have those knowledge, we try to apply those knowledge in the biological systems. Okay, that is the biotechnology. So principles of biotechnology, what we do, you can think of what is actually any organisms. Okay, any organism may be cellular, unicellular or multicellular, or having organ system. Okay, like our human species or some other animals also, like mammalian or some other organisms, some big animals. Okay, we have organ system level of organization, right? Means many organs are there, they are acting together to facilitate the process of living, right? So but in case of unicellular, only cellular level organization, means only cellular components are there, right? Many organelles are there, organelles inside the cell. That organelles are working together to give that life processes, to give that life forms in unicellular level. Likewise multicellular, multiple cells are there for controlling and enhancing and facilitating the life processes. Okay, so so genetic material, you people know, I think, right? Genetic material means whatever we have in the cell, right? We have, or any organism have inside the cell, that is that control, that regulate, that control the expression, that regulates the life processes of that organism. Okay, so that is the genetic material. And what happens when one organism, from one organism another organism is formed, then the genetic material of the parental organism is transferred to the progeny cells or progeny organisms, right? So what is the genetic material is controlling function is there. So controlling function is almost same, because same material is transferred from the parental organism to the progeny cell, right, or progeny organism. That's why. So what we can do, if we change, now think of it, if we change that genetic material, so in some forms, so what could be the result? The genetic makeup will be changed, and thus if as we already know, that genetic makeup is controlling or regulating the life processes, or the nature of the life of that organism, or the consciousness or the respondingness of that organism. So if we change the genetic material or genetic makeup, eventually the expression, the nature, the consciousness, or the life processes of the organism will also get changed, right? That's why genetic engineering, this is the genetic engineering means changing. Okay, genetic engineering simply we are trying to change the genetic makeup, we are changing the genetic content. So think another aspect, like in some organism the genetic makeup is deformed, already deformed, maybe because of mutation means sudden change of the genetic material. Okay, so maybe due to mutation, maybe UV exposure, okay, x-ray exposure, many reasons are there. Reason could be anything, but what happened ultimately, the genetic makeup got changed. So now if we try to make that right one again, then what is happening, deformed genetic material is now converted to the actual form or ideal form, or the previous form, so it is the correct one. So by this we can correct also genetic, if genetic problem is there, means genetic makeup is changed by somehow, by some means, then we can try also to correct that. Okay, so this is the genetic engineering. Okay, so obviously DNA, RNA, I will discuss it later. DNA and RNA, deoxyribonucleic acid, and RNA means ribonucleic acid. So DNA and RNA both are present inside our body system. In some organisms, in unicellular organisms, only RNA is present. Maybe in other organism only DNA is present. Okay, and in case of retrovirus, this is the example of retrovirus, HIV virus. Okay, HIV virus is a form of retrovirus. What happens in that organism, actually RNA is present inside the organism usually in the usual case or normal time. But what happens sometimes when these retrovirus get incorporated inside our body system, means our body. Okay, HIV enters inside our body, then this RNA is converted to DNA. This is the process known as uh, reverse transcription, because DNA to RNA conversion is known as transcription. So now what happened, RNA to DNA is conversion occurred, so this is the reverse transcription, and the enzyme involved in this process is known as reverse transcriptase. So reverse transcriptase enzyme is present inside retroviruses. Okay, and the example is HIV virus. Okay, that causes AIDS. Okay, okay, next. So okay, so these are the things, recombinant DNA, gene cloning, I will cover one by one. Okay, so see, recombinant DNA actually genetic material, maybe RNA or DNA. If we consider that the an organism is having DNA as a genetic material, so deoxyribonucleic acid, this is a polymer form of nucleic acid. Okay, deoxyribonucleic acid. Now what we can do, earlier I said that in genetic engineering we try to make it right one if it is already deformed, or else we can in some cases we try to change the DNA makeup, DNA content, to get our desired characteristic, means actually the characteristic feature or the nature or the expression of that organism is controlled by DNA. So now what we are doing, the changing the genetic makeup or the DNA content. So DNA content changing means the characteristic feature will also get changed. That's why recombinant DNA means whenever we changed DNA, means part of DNA is known as gene, I will discuss it later. Okay, gene, the characteristic unit, that part gene is the unit of the genetics, means many genes are there inside the DNA. One gene controlling one character, usually in some cases many gene controls one character, it also happens. And another thing happens, one gene controlling or involved in many characters, that also happens. So in normal cases or for discussion, we can assume that one gene is controlling one character only. So when we change that gene, we incorporating another gene in that place, so character will get changed. That is, whatever we changed, whatever the changes we made in the DNA, that is known as the recombinant DNA. Done. Okay, so gene cloning means cloning is the replication or the copy, making of another copy of the same thing. Maybe like you can imagine one cell, we if we made same type of cell, same genetic material, the cell having same genetic material, and everything is same, then only we can say that cell cloning is done. Means both cell are same only, everything in every aspect. Likewise, if we can multiple a specific gene in some processes, we have those processes. Okay, I will discuss later. So that is known as gene cloning. Okay, so gene transfer means we are incorporating some foreign gene, means organism was not having that gene. Now from outside we are incorporating that foreign gene inside the DNA. That is known as a gene transfer. Okay, so plasmid, very important thing. Plasmid means we know E. coli, right? Escherichia coli bacteria. That bacteria hot is there inside that cell. In case of Escherichia coli, the DNA is the genetic material. DNA acts as genetic material. Out besides this, some genetic material is also present inside the cell of Escherichia coli. That is known as the plasmid. So it is known as the autonomously. Okay, no one controlling that plasmid, autonomously replicating. It can produce another another quantity of DNA. Okay, so replicating circular circular, it is in the circular form, extra chromosomal DNA. So chromosomal DNA means genetic control, genetic regulation is maintained by that part. But extra chromosomal outside, besides the chromosomal DNA, that is the plasmid. It is present inside the Escherichia coli, okay, or E. coli. Okay, so okay, these are the basic things I have discussed. I will discuss in detail in other lectures. Okay, thank you.